Rabies virus is a negative sense RNA virus that affects the central nervous system, such as the brain and spinal cord, causing inflammation. It is a type of species of the genus Lysovirus within the Rabidoviiridae family, which are negative, single-stranded RNA viruses with a distinctive bullet-shaped structure. It is a zoonotic disease, meaning it is passed on from animals to humans. Because it is a nootropic that sheds in saliva, animals pass it to humans when they come in contact via a bite, which is the most common way of transmission. Any mammal can get rabies, but the main animals in the US that exhibit rabies are foxes, skunks, raccoons, and bats. Rabies virus can be categorized as either fixed or street wild type. The differences in the strain correlate to how effective they attack and evade the host's immune system. Clinical manifestations of rabies is characterized by general malaise, fever, fatigue. Respiratory system symptoms include sore throat, cough, and shortness of breath. Gastrointestinal symptoms include anorexia, dysphagia, nausea, vomiting, abdominal pain, and diarrhea. Central nervous system symptoms include headache, vertigo, anxiety, apprehension, irritability, and nervousness. Other remarkable abnormalities include agitation, photophobia, priapism, increased libido, insomnia, nightmares, and depression. Another interesting symptom created by rabies is hydrophobia, which is the adverse behavior towards water or water consumption. This is one of the reasons why we see patients foam at the mouth. Infected salivary glands cause overproduction of saliva. This, coupled with muscle spasms in the throat and voice box, makes swallowing difficult and creates foaming at the mouth. There are 5 stages of rabies, the first being the incubation period, the period between exposure to infection and the appearance of first symptoms. This usually takes 30 to 90 days, but can range from 5 days to longer than 2 years after initial exposure. Variability in the length of incubation period depends on several factors. For example, incubation periods are shorter in children and individuals being closer to the central nervous system, such as the head. Size of the viral inoculum also plays a role, as well as the length of the neural path from wound to brain, and the strain of the rabies virus. Next is the prodrome period, where early symptoms indicating the onset of disease or illness occur. Clinical symptoms are first noted during this period, which lasts 2 to 10 days. If the patient is not vaccinated or treated before this period, the prognosis is usually always fatal, ending in death. According to the antiviral research, the fatality rate of this disease is almost 100%. The next period is called the acute neurological period, which begins with objective signs of central nervous system dysfunction. The disease is classified as furious rabies if hyperactivities such as hydrophobia predominates, and dumb rabies if paralysis dominates the clinical picture. In both forms, however, fever, paresthesia, muscle fasciculations, hyperventilation, and hypersalivation may occur. At the end of the acute neurological phase, periods of rapid, irregular breathing may begin, followed by paralysis, coma, and then death. Entry of the virus into the host happens via injection into muscle tissue or other peripheral tissues. After the virus attaches to the host cell membranes via the G protein, it penetrates the cytoplasm by fusion or pinocytosis, the introduction of fluids into a cell by invagination of the cell membrane, followed by formation of vesicles within the cells. Fusion of the rabies virus envelope to the host cell membrane starts the infection process. Primary transcription is then initiated, where each RNA is translated into individual viral proteins and positive-stranded RNA, which acts as a template for the production of progeny negative-stranded RNA. The rabies virus undergoes low levels of replication in the cytoplasm of infected tissue that does not spread in the muscle tissue. This period of low replication without spread explains the variable incubation period of the virus during infection. Because of this, the rabies virus is able to later gain entry into the peripheral nervous system and subsequently into the central nervous system. Entry into the peripheral nervous system occurs when it infects the motor end plates of muscle tissues and enters via neuromuscular junctions. The G protein component of the virus binds to the nicotinic acetylcholine receptors at the neuromuscular junction to gain access into the peripheral nervous system, which will then travel within axons in peripheral nerves by retrograde axonal transport. The rabies virus possesses proteins that plays a role in sequestering the immune response, which allows it to efficiently replicate in muscle tissues before moving on to infect the peripheral and central nervous systems. For this reason, the virus is extremely neuroinvasive. Sequestration is primarily dependent on the P protein, N protein, and G protein components, which specifically targets and decreases expression of interferon beta, myoxovirus resistance protein 1, and oligoadenylate synthase 1 genes that are important in innate responses against viral infections. The virus's ability to evade the initial immune response in the muscle and enter the peripheral and central nervous system is dependent on the specific strain, with more pathogenic strains exhibiting an increased efficiency for neuroinvasiveness. The first line of defense against viral infections is type 1 interferon. Type 1 interferon activates the JAK-STAT intracellular signaling pathway, leading to transcriptional induction of hundreds of IFN-stimulated genes, many of which have antiviral activities against a wide range of viruses. However, the rabies virus has evolved different strategies to counteract the antiviral effects of type 1 interferon responses. The P protein on rabies inhibits interferon production by impairing interferon regulatory factor 3, preventing antiviral genes from being activated. For example, rabies is able to evade initial immune responses in the muscle by impeding the dendritic cell's autocrine feedback loop. The P protein expressed by the virus has been shown to inhibit the JAK-STAT signaling cascade, which is responsible for upregulating the production of proteins required for the autocrine feedback loop of interferon alpha and beta. If the autocrine feedback loop of interferon alpha and beta is not produced, then the dendritic cells cannot undergo maturation. In the absence of interferon alpha and beta, viral replication in the dendritic cells occurs at a more rapid rate, thus increasing the pathogenicity of the virus. The rabies virus's N protein component has been reported to play an important role in the evasion of host RIG-1-mediated antiviral responses by inhibiting genes that upregulates RIG-1-like receptors, or RLRs, which is a pattern recognition receptor that is useful for detecting viral pathogens and thereby allowing the virus to propagate efficiently and spread quickly. The rabies virus is also able to evade T-cell attacks because it has mechanisms in place that decreases the amount of T cells. It does this during the replication process by using evasive strategies, similar to that of tumor cells. It upregulates surface molecules FAS-L and PD-1 to trigger apoptosis in T cells, and decreases the production of cytokines, reducing the effectiveness of the immune system cells from signaling one another. Furthermore, T cells that infiltrate the nervous system to attack the rabies virus are downregulated due to several neuropeptides and neurotransmitters. It is both interesting and scary to know that the rabies virus specifically targets the central nervous system, where the immune system cannot operate robustly. The rabies virus defends against natural killer cells by upregulating HLA-E, which stands for Human Leukocyte Antigen Class 1 Histocompatibility Antigen Alpha Chain E, which is a protein that induces apoptosis in natural killer cells. Once the virus reaches the motor neurons of the spinal cord, it begins to rapidly ascend to infect the brain and will then continue to spread to the salivary glands, skin, cornea, as well as other organs.
The prognosis for rabies is usually always fatal, ending in death. Luckily, rabies shots can be obtained through passive protection. During this duration of time, the shot is injected near the area that has been infected. The immunoglobulin, or more known as antibodies, make proteins to help fight against antigens. During the course of the treatment, the patient will receive 4 doses of vaccinations during a 14-day period. Another key step is prevention. This vaccination generally occurs to those who have higher exposure to rabid animals. 3 doses will be taken in a time frame of 28 days. However, it does not guarantee you're immune to rabies. If bitten, you should still seek help from a doctor in which you are infected. They will give you the previously 2 dosage on day 1 and 3 without needing to obtain the immunoglobulin shot, in which the body should resolve itself. So what are some things to look at to identify if the animal you encounter have rabies? One way is by looking at the overall health of the animal. If the animal is acting mad or strange, chances are the animal is rabid and you should seek help. Another way, if possible, is to safely obtain the animal. as it can be tested in lab to see if it has contracted rabies by looking at the brain. Therefore, even if it is by self-defense, try to avoid using blunt force on the animal's head as a way to capture it. In the meantime, if severely injured, you should stop the bleeding by applying pressure and washing the wound with soap and water. A famous treatment has been a key role factor in rabies is called the Milwaukee case. The occurrence of a 15-year-old girl who had developed rabies after being exposed to a bat was induced into a coma using an antiviral cocktail. The coma was to suppress her brain activity where it would minimize the damage to her immune system. This took place at the Milwaukee Hospital, hence the name, and in which she survived this illness with mild neurological consequences. The girl has not been vaccinated at that time, and because of the success, it inspired similar therapies that were carried out but resulted in failure. Potential effects include immunosuppression from barbiturate, metolazam, ketamine, and ribavirin. In the future, to improve therapy, scientists aim to search for a good baseline or a normal functional immune system. The concept of being vaccinated before getting sick and focus more on bats rather than canines. In addition to this, scientists would like to neutralize antibodies in serum with additional CSF and to have mild therapy. The mild therapy will use ribavirin and interferon alpha to help fight against the rabies. The function of ribavirin inhibits the virus in vitro, while the interferon alpha cytokines are effective in monkeys, but not to patients with high doses. The therapy aims for the penetration in blood barriers, because it is in the CNS infection that limits both antiviral agents. The additional ketamine will inhibit the infection in the brain regions of transcription. Overall, the main objective is to prevent neuronal damage in humans and also to have more of an effective therapy.
The CDC reports that rabid dogs are responsible for 90% of human exposure to rabies and 99% of rabies deaths worldwide. Though the vaccines to prevent human rabies have been available for over 100 years, they are costly to distribute and implement. In the United States alone, over $300 million each year is spent on rabies prevention, most of which is dog vaccination, the most direct way to mitigate the disease. Because of this, countries affected worst by rabies tend to be poorer countries with inadequate access to health care and preventative measures, with little to no rabies surveillance to speak of. There is also the issue of population turnover. The annual turnover for dog populations is 25%, which would require annual vaccinations to control the virus, costing even more money. Not to mention that perfect control of the populations is difficult to obtain, and the possibility of the disease escaping and reintroducing into controlled areas is very high. Wildlife reserves in Europe and North America also protect the wild reservoir of the rabies virus, which could continue to pose the threat of reintroduction as well. Other organizations like the WHO, OIE, World Organization for Animal Health, and FAO, Food and Agriculture Organization of the United States, have all pledged to help end rabies infection by dog bites by 2030. Globally, the main funding for rabies studies comes from the CDC and from the Global Alliance for Rabies Control. Both of these conduct their own research into understanding rabies better and better, more accessible treatment options for it. In addition, they work to raise awareness about rabies and help distribute donations of rabies vaccines. On September 12, 2018, a donation of 75,000 doses of rabies vaccine was made to GARC, the Global Alliance for Rabies Control, to treat dogs in Madagascar in an attempt to control the disease and mitigate its effects. In addition, there are many localized foundations for rabies research and prevention, including Rabies in the Americas and Rabies in Asia. September 28th has also been the day to observe Rabies Awareness Day since 2007 in an effort to bring light to the subject on an international scale. Global awareness of the misunderstandings of rabies is an important step. Rabies is not an inherently human disease. Humans incidentally catch it from infected animals. Underreporting of the virus is already an issue, but reporting the populations of animals infected and working to understand them would be more helpful for treating the problem at the source. All of this research and awareness would help save and treat the 59,000 people who die each year of a completely preventable disease.